Alright, in this video we're going to just be basically reviewing the most important parts of mitosis and doing a little bit of a self-quizzing to see if we completely understood what we need to understand. So remember that the interphase is the major part of the cell. The cell will spend about 90% of its life on the interphase. And it's actually divided into the G1, S, and G2 phases. Now, in all of these three stages, the cell will continue to do its normal metabolic activities, including anabolic and catabolic processes such as building proteins, digestion, constructing fat and carbohydrates, polysaccharides, membrane transport, cell respiration, photosynthesis, all of these things. But during the G1 phase, in addition to all of that, it's also growing following the cell division. We're going from a small daughter cell size to normal cell size. And it's also replicating organelles to a normal amount of organelles that the cell should have, not just half of the organelles, which is usually what it comes out after division takes place. Also remember that during the S phase, the cell also, in addition to normal metabolic activities, doubles the DNA. And from that point on, the cell actually has twice as much DNA as usual. And then at the G2 phase, the cell prepares for division by storing energy, initiating chemical reactions, and doubling a lot of the organelles as well. Now at the end of the G2 phase, the cell initiates mitosis, and mitosis actually happens in 4 stages. Now, the prophase is all about packaging the DNA and unassembling the nucleus to facilitate the actual duplication process. Remember that mitosis is about duplicating the nucleus, while cell division in general is just like cytokinesis or binary fission, it's just splitting the cell in half. So karyokinesis or mitosis is about splitting the nucleus, which is something that's peculiar to eukaryotes. During the metaphase, what's peculiar about it is that the cells are lined up in the equator of the cell. There's a formation of the poles with the asters, the centrosomes, and centrioles in animal cells only. And you see the full formed spindle, which is ready to separate the cells. Metaphase is all about lining up the cells in a format that will allow the chromatids to separate successively in a way that each daughter cell receives exactly the same half copy of what's now doubled the DNA. So that way each daughter cell will be identical to its mother cell. Anaphase is the actual separation, where the enzymes cleave the centromere and also depolarize the aster fibers, shortening the fibers, while the centrioles pull them towards the poles, creating a separation. As soon as those chromatids reach the poles, they start to uncoil during the telophase, and where the nucleolus and the nuclear envelope will reform after having deformed during the prophase, but this time it will happen twice, each on the opposite ends of the cell. And towards the very end of telophase, a separate non-mitotic process, which sometimes is included in mitosis, but it's really basically just the cell division itself, and that is called cytokinesis. In this case, you see a cleavage furrow, which therefore must be an animal cell. So this will be the metaphase plate, that will be the cleavage furrow, all right, to answer the questions that are in the drawing. Now that you've done this review, you should be able to answer these questions. Where is each of the cycles starting here? So number 1 must be interphase, because I see nucleoluses in fully normal chromatin with the uncoiled DNA, which just looks cloudy. But on 2, I start to see cloudiness, so maybe that's an early prophase. By 3, I don't see a nuclear envelope anymore. The nucleolus is almost gone, so that might be late prophase. This is prometaphase, fully developed nucleolus, so it might still be there. So it might be towards the late of the prophase as well. By 5, we actually hit the end of prophase. Nucleolus is gone, nuclear envelope is gone. All you see is these condensed chromosomes, which will now start to be connected to the kinetochore fibers and sent towards the polar metaphase plate, which is what you see in 6. So 6 will be metaphase. On 7, you see this beginning of anaphase, which concludes in 8. And by 9, you see the telophase, where they are already at the separate ends and will start to uncoil, which is what you see at 10. And this time you see a cell plate, because we're talking about cytokinesis at the plant cell. So you can actually see the sequence taking place here. And remember that the anaphase would be the shortest phase, followed by the metaphase, followed by telophase, and then the prophase will be the longest phase. Now all of these phases are about 10% of the cycle altogether, and 90% of the cycle is a number 1, which is the interphase, where the cell performs normal metabolic activities. Alright, now let's see if you can continue this and figure out what these are. What do you think the first one is? Alright, I see that the chromatids are already separated. That means it must be anaphase, okay? And early anaphase in that case. Now here I see a nucleolus. I see the nuclear envelope starting to go away, and maybe a few condensation here and there, perhaps early prophase. On this here, I see the spindle starting to form. Chromatin is even more condensed. I don't see the nucleolus anywhere. This is going to be a middle to late prophase. On the third one, I see things lined up in the equator, a metaphase plate, the poles form, a full spindle. This must be metaphase. This will be telophase because I actually start to see the cleavage furrow forming, and I also start to see the formation of the nucleus on the opposite ends of the cells. This right here, I see chromosomes, but I do not see a fully formed spindle or the metaphase plate. I would say the late prophase, extremely late prophase, so play me pro-metaphase. This here would be very late telophase because I actually see a fully formed nucleus on the opposite end. So this would be like late telophase or very late cytokinesis. The cell's about to divide. Again here, maybe perhaps prophase because I see the condensing, the spindle's not formed yet, and I still even see a hint of the nuclear envelope. This, on the other hand, I see chromatids separate, but I still see a piece of the spindle, so it can't be telophase, so this must be anaphase as well. So you see how you can figure things out, and hopefully this prepares you to do this part, which is finding the phases or stages in this picture. Now, I definitely see the prophase right here. This has got to be prophase because I see the consolidation going away. This might also be early prophase. The other ones will be probably be our anaphase. This might be late telophase right here because the cells are small. So this might be a cell plate almost completely formed. So this might be late telophase, cytokinesis as well. If I had to bet on metaphase, I would say this one would be my metaphase. Actually see the spindle here. You get hints of the spindle everywhere. This one and perhaps this one would be most likely anaphases because I see a fully formed spindle. I don't see a fully formed spindle here, so it would either be perhaps a prophase, late prophase, okay? Let's see what else I spot in there. Maybe a prophase over here because I see the formation of the chromosomes on the top. All of the other ones will probably be interphase. Maybe this one will start to be a little bit of prophase, but all the other ones will probably be around interphase. So, as you can see, the majority of cells are in interphase, which was just normal. Barely any cells in anaphase or metaphase, and telophase is also rare because it's just hard to identify it. All right, so this is what you're going to have to do when you do the lab on mitosis in the class. So you should practice looking at these on the virtual mitosis lab, and you see if you can figure it out how to do this. All right, here again, can you find it? Definitely metaphase here in the middle, right? Here's some prophase right there. If you see a nucleolus, automatically interphase. So all of these with the nucleolus inside and the shady chromatin is going to be interphase. All right. So these are just some examples of cell plates, and that you're going to experience as well in class to try to figure out the cell cycle. All right. Now, last thing I want to do, speaking about the mitosis, is what happens when mitosis goes out of control. So, mitosis, in a separate video, we're going to talk about cell control, the control of the cell cycle. When mitosis goes out of control and the cells just duplicate out of control, instead of getting a normal cell, you get these cancer cells which take over the tissue, sometimes secrete toxins, and might actually cause damage to the surrounding tissue, and sometimes even spread all over the body and become metastatic or undergo metastasis, which is the really bad types of what are we talking about? Cancer. Now, tumors are not always malign. To be malign, they have to be either damaging the tissue or secreting toxins which are causing damage to the tissue, or either displacing the tissue by replacing it with these bad cells. But sometimes benign tumors will grow and not cause problems for the immediate environment, although they sometimes cause pressure, which is a problem, or can also develop into malignant tumors. So usually people remove benign tumors just to be safe. But cancer is basically an event where the cell cycle is not controlled properly. And you see a picture of a representation of a cancer cell on the right top hand side corner. Actual pictures of cancer cells on the tissue that this should not have them on the top left corner. And then an actual picture of scanned cancer as an example of non-native tissue which is growing out of control. Now, cancer has a lot of problems. Typically, you have production of toxic proteins, you have an uncontrollable cell cycle, you have uncontrollable cell growth, you also have the production of several proteins which cause the cell to have accelerated growth, and you also have less control over a correction of mutations in the cell. When all of these things are put together, that's why cells undergo cancer. Now, normally, when a cell gets to this stage, it kills itself through a process called apoptosis before it gets this far. And even if the apoptosis fails, by the way, apoptosis happens through the lysosomes attacking the cell from the inside out. The cells are programmed for that. But when that fails, maybe because of mutation, which is, again, common in cancer cells, this body itself tries to attack the cancer cells with its immune system. So you're only going to get cancer if you actually have too many mutations, if you're not healthy enough, your immune system is unbalanced, you don't have a proper diet or proper rest, or if you're exposed to radiation or mutagens or any carcinogens or other chemicals which could be causing cancer. So it's a combination of things which are actually going to lead to actual cancer. But remember that it's a series of mutations that actually leads to this place. But one very important fact, that is the first one we talked about this year, is the fact that the uncontrollable cell growth, or mitosis. All right. So, for example, here you see on the right side the normal cells, when they touch each other, that sends a cell signal that deactivates cell growth by inhibition by touch. But cancer cells do not listen to that cell signal and start mounting on top of each other, which is what actually helps develop tumors. So yet another example of a mutation or a problem that cancer cells usually have. And we talk more about that when we do the cell regulation or cell communication lecture. But either way, understand at this point that the cancer cells are basically a product of uncontrollable cell growth, among other problems. All right? Now, and remember also that if cells actually grow faster than the immune system can kill them, that's when you're actually going to get the cancer. Now, the last thing, I want to talk about differences between plant and animal cell mitosis. On the top right corner, you see a structure made of microtubules as a series of several microtubules, triplets surrounded in circles, and they always show up in pairs. These are the centrioles, and they're produced by centrosomes in animal cells only. Plant cells still have centrosomes, but they don't make centrioles. Centrioles help create the spindle and accelerate the cell division process, which is why it's usually faster in animal cells. Another big difference between animal cells and plant cells is that plant cells divide by cell plate because they have the cell wall which impedes them from imploding inwards the way the animal cells do through the cleavage furrow. So instead, plant cells grow a cell plate from the inside out and grow a new cell wall to separate the two new cells during the cytokinesis. But animal cells do it through the cleavage furrow, which is an infolding of the cell membrane, as you can see that. And other than that, the cell cycle pretty much looks the same. So you can see here the comparison between onion root mitosis, which is a plant, and whitefish blastula, which is the embryo of a whitefish. So as you can see here, it looks very similar in terms of the actual structures. So basically, it's basically the dedicated organelle, which is the centriole. The cytokinesis is slightly different, but otherwise it's pretty much the same, including the fibers, which are made of cytoskeletal materials of microtubules. And the process is also pretty much similar. All right, so that's a roundup of mitosis, and I hope it was helpful to you. And we'll go out from here to talk about meiosis.